Rain has died down a little bit, but it's still the chilly, chilly wind and the noise. The buses and the cars and the trucks keep coming by, but this is my joy to go and release. This is the best I can offer for the day, and when the weather gets better and this virus thing goes away, maybe I'll still be here and I can keep doing this a lot clearer in a silent room there that I had at the library. Important notes. These notes are about the blood clotting mechanism that has been shown to be boringly, irreducibly complex. And this is a microbiologist who's saving us from further detail. It's just absolutely overwhelming, the amount of stuff that has to happen all of it at the same time. The system has to be completed all at once. If one item of this system is missing, the entire mechanism fails, and the living being, whether it be a rat or a human being or a cow or whatever, they die. The system has to work over and over again precisely. So, Michael Behe summarizes important notes. Most importantly, the clotting cascade has to be turned off at some point before the organism completely solidifies. And that means it dies. The blood clotting system fits the definition of irreducible complexity. That is, it is a single system composed of several interacting parts, that contribute to the basic function, and where the removal of any of the parts causes the system effectively to cease functioning. The function of the blood clotting system is to form a solid barrier at the right time and place that is able to stop blood flow out of an injured vessel. The components of the system, beyond the fork in the pathway, are fibrinogen, prothrombin, Stuart factor, and proaccelerin. None of the cascade proteins are used for anything except controlling the formation of a blood clot. Yet in the absence of any one of the components, blood does not clot and the system fails. There are other ways to stop blood flow from wounds, but those ways are not step-by-step precursors to the clotting cascade. For example, the body can constrict blood vessels near a cut to help staunch blood flow. Also, blood cells called platelets stick to the area around a cut, helping to plug small wounds. But those systems cannot be transformed gradually into the blood clotting system, for none of the components closely resembles those of the cascading system and would have problems of their own in their transformation, for they too are irreducibly complex when all interactive components are considered. Just consider the heart, lungs, the brain, the function of the different organs, all have to work together. One can block the other from functioning, and the living being is dead. The simplest blood clotting system imaginable might be just a single protein that randomly aggregated when the organism was cut. The simplest clotting system would be triggered inappropriately, causing random damage and wasting resources, would not meet the criterion of minimal function. It's just not the final activity that is the problem. Rather, it is the control system. Turn it on, turn it off. How much, how little. One could imagine a blood clotting system that was somewhat simpler than the real one, where, say, Stuart factor, after activation by the rest of the cascade, directly cuts fibrinogen to form fibrin, bypassing thrombin, leaving aside for the moment issues of control and timing of clot formation. Upon reflection, we can quickly see that even such a slightly simplified system cannot change gradually into the more complex intact system. If a new protein were inserted into the thrombone-less system, it would either turn the system on immediately, resulting in rapid death, or it would do nothing, and so have no reason to be selected. Because of the nature of a cascade, a new protein would immediately have to be regulated. From the beginning, a new step in the cascade would require both a coenzyme and also an activated enzyme to switch on the coenzyme at the correct time and place. Since each step necessarily requires several parts, not only is the entire blood clotting system irreducibly complex, but so is each step in the pathway. Each of the control points of the blood clotting cascade Each of the control points of the blood clotting cascade needs both an inactive proenzyme and a separate enzyme to activate it. Point taken. Evolution does not have a chance to explain how the blood coagulating system works. But he says, is it possible that this ultra-complex system could have evolved according to Darwinian theory? The observation of split genes, wherein similarities were observed in the genetic structures of different parts of biological systems, led to the hypothesis that perhaps new proteins could be made by shuffling the DNA fragments of genes that code for parts of old proteins, much as cards can be picked from several piles to give a new arrangement. To support the hypotheses, advocates point to similarities in the amino acid sequences and shapes of discrete portions called domains of different proteins. The proteins of the blood coagulation cascade are often used as evidence for shuffling. Some regions of cascade proteins coded by separate gene pieces have similarities in their amino acid sequences with other regions of the same protein. That is, they are self-similar. Also, there are similarities between regions of different proteins of the cascade. For example, proconvertin, Christmas factor, Stuart factor, and prothrombin all have a roughly similar region of their amino acid sequences. Additionally, in all those proteins, the sequence is modified by vitamin K. Furthermore, the regions are similar in sequence to other proteins not involved with blood coagulation at all, but are also modified by vitamin K. The sequence similarities are there for all to see and cannot be disputed. By itself, however, the hypothesis of gene duplication and shuffling says nothing about how any particular protein or protein system was first produced, whether slowly or suddenly, or whether by natural selection or some other mechanism. Remember, a mousetrap spring might in some way resemble a clock spring, and a crowbar might resemble a mousetrap hammer, but the similarities say nothing about how a mousetrap is produced. In order to claim that system developed gradually by a Darwinian mechanism, a person must show that the function of the system could have been formed by numerous successive slight modifications within an acceptable time frame. Of course, as we look at it, the probability of that is beyond the age of the universe. Just for one system within one living being. The explanation is seriously inadequate because no reasons are given for the appearance of the proteins. No attempt is made to calculate the probability of the protein's appearance, and no attempt is made to estimate the new protein's properties. The first thing to notice is that no causative factors in the evolutionary models proposed are cited. Thus tissue factor appears, fibrinogen is born, antiplasmin arises, TPA springs forth, a cross-linking protein is unleashed, and so forth. What exactly, we might ask, is causing all of this springing and unleashing in such a precise manner, and then unleashing when it's time to stop the blood clotting? A step-by-step Darwinian scenario involving the undirected random duplication and recombination of gene pieces is in view. But consider the enormous amount of luck needed to get the right gene pieces in the right place at precisely the right time. Eukaryotic organisms have quite a few gene pieces, and apparently the process that switches them is random. So making a new blood coagulation protein by shuffling it is like picking a dozen sentences randomly from an encyclopedia in the hope of making a coherent paragraph. Furthermore, the Darwinian scenario typically does not go to the trouble of calculating how many incorrect, inactive, useless, variously shuffled domains would have to be discarded before obtaining a protein with, say, TPA-like activity. To illustrate the problem, let's do our own quick calculation. Consider that animals with blood clotting cascades have roughly 10,000 genes, each of which is divided into an average of 3 pieces. This gives a total of about 30,000 gene pieces. TPA has 4 different types of domains. By variously shuffling, the odds of getting those 4 domains together is 30,000 to the 4th power, which is approximately 1/10 to the 18th power. Now, if the Irish sweepstakes had odds of winning of 1/10 to the 18th power, and if 1 million people played the lottery each year, it would take an average of about 1,000 billion years before anyone, not just a particular person, won the lottery. What's 1,000 billion years? That's a little older than the universe, you'd think. 1,000 billion years is roughly 100 times the current estimate of the age of the universe. The same problem of ultra-slim odds would trouble the appearance of prothrombin, fibrinogen, plasminogen, proaccelerin, and each of the several proposed rearrangements of prothrombin. Unfortunately, the universe doesn't have time to wait. So much for that. The second question to consider is the implicit assumption that a protein made from a duplicated gene would immediately have the new necessary properties. Thus, we are told by the Darwinian model type, tissue factor appears as the result of the duplication of a gene for another protein. But tissue factor would certainly not appear as the result of the duplication. The other protein would. If a factor for making bicycles were duplicated, it would make bicycles, not motorcycles. That's what it meant by the word duplication. A gene for a protein might be duplicated by a random mutation, but it does not just happen to also have sophisticated new properties. Since a duplicated gene is simply a copy of the old gene, an explanation for the appearance of tissue factor must include the putative route it took to acquire a new function. This problem is discreetly avoided. The Darwinian model runs into the same problem in the production of prothrombin, a thrombin receptor, antithrombin, plasminogen, antiplasmin, proaccelerin, Stuart factor, proconvertin, Christmas factor, antihemophilic factor, and protein C. Virtually every protein of the system. Far longer time would it take than the universe is permitted by evolutionists to be old. The third problem in the blood coagulation scenario is that it avoids the crucial issues of how much, how fast, when, and where. Nothing is said about the amount of clotting material initially available, the strength of the clot that would be formed by a primitive system, the length of time the clot would take to form once a cut occurred, what fluid pressure the clot would resist, how detrimental the formation of inappropriate clots would be, or 100 other such questions. The absolute and relative values of these factors and others could make any particular hypothetical system either possible or much more likely wildly wrong. And you got the species dies out for the umpteenth time. For example, if only a small amount of fibrinogen were available, it would not cover a wound. If a primitive fibrin formed a random blob instead of a meshwork, it would be unlikely to stop blood flow. If the initial action of antithrombin were too fast, the initial action of thrombin too slow, or the original Stuart factor or Christmas factor or hemophilic factor bound too loosely or too tightly, or if they bound to the inactive forms of their targets as well as the active forms, then the whole system would crash. At no step, not even one, does any of the Darwinian models give a model that includes numbers or quantities. Without numbers, there is no science. When a merely verbal picture is painted of the development of such a complex system, there's absolutely no way to know if it would actually work. When such crucial questions are ignored, we leave science. Yet the objections raised so far are not the most serious. Wow. The most serious, and perhaps the most obvious, contains irreducible complexity. I emphasize that natural selection, the engine of Darwinian evolution, only works if there is something to select, something that is useful right now, not in the future. Suppose, for purposes of discussion, as might occur in a typical Darwinian model, no blood clotting appears until at least the third step. The formation of tissue factor at the first step is thus unexplained, since it would then be sitting around with nothing to do. In the next step, prothrombin, popping up already endowed with the ability to bind tissue factor, which somehow activates it, the poor proto-prothrombin would also be twiddling its thumbs with nothing to do until at last a hypothetical thrombin receptor appears at the third step and fibrinogen falls from heaven at step 4. These are magical things, not evolutionary things. Plasminogen appears in one step. We'll more on this later.